Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the third webinar in the Women's Health 2024 series. Today's webinar is titled Effects of Ovarian Failure on Muscle Form and Function, featuring Paris Mashuri, a PhD candidate in the Department of Human Health and Nutritional Sciences in the Neuromechanical Performance Research Lab at the University of Guelph. Today, she'll dive into her pioneering research on the effects of gradual ovarian failure on skeletal muscle function. Before we get started, we would just like to acknowledge our partners for the series, the American Physiological Society, the Society for Women's Health Research, and Scientists.com. We'd also like to say thank you and give a shout out to all of our series sponsors. None of this would be possible without them. We'd like to give a special shout out today to our session sponsor, Aurora Scientific. For over 30 years, Aurora Scientific measurement instrumentation has been the choice of leading science researchers around the world. Their selection of dual-mode lever systems and force transducers are used in a broad array of life science research specialties, including exercise and metabolism, cardiology, injury recovery, gerontology, disease progression, pharmacology, and so much more. Whether measuring force in single myofibrils or quantifying the mechanical properties of whole muscle, Aurora Scientific has the expertise and the instruments to support the most demanding of muscle physiology experiments. So a huge thank you again to them. I'm Sydney Otamanchek from the events team at Scientists.com, and I'm happy to be your host for today's event. Before I welcome our speaker to the floor, I'd like to cover a few housekeeping notes to help you all get the most out of your experience today. First, you can take a look at the interactivity panel at the right of your screen with 4 tabs: Chat, Q&A, Polls, and Handouts. We want this to be an interactive experience for you, so please send in your questions, thoughts, and comments using the Q&A tab. We will try to get to as many of your questions as we can at the end of the webinar today. When a question is added into the Q&A panel, you can upvote the question to increase its position on my list by clicking a thumbs up button underneath it. This is always helpful so I know which questions to address first. You can also explore some links and documents in the Handouts tab to learn more. Lastly, if you're having any technical issues, please first try to refresh your browser and ensure you're using Google Chrome to access this event, as it is the most stable browser. If you're still having any trouble after you've refreshed, you can use the private chat function to communicate with me by sending in a chat to Sydney, and I'll be happy to help you troubleshoot. Okay, so before we get started, I'd just like to run a couple of quick audience polls to get to know you all a little bit better this morning. So our first question is, what are the greatest perceived challenges to implementing muscle mechanics or contractile research techniques in your lab? And this is a select all that applies. So our options this morning are lack of proper direction from literature, lack of in-house expertise or resources, lack of training or training programs, perhaps the expense of the equipment. If this is not relevant to your research, go ahead and select option E. Or perhaps you've already implemented these techniques in your lab, so you can select option F for that one. And I'll just give you all a moment to complete that poll for us. Wonderful. We already have a lot of answers coming in. And the poll will stay open for the entirety of the talk. So if you'd like some time to think about your answer, you can skip it for now and always go back to the polls tab to send in your response later on. Okay. And our second poll, how much experience do you have with muscle mechanics or contractility measurements? So we just want to know what your experience level is here. So whether you have extensive experience, only some, perhaps understand the principles, but you don't have any experience. And if you have no experience at all, just let us know with option D. And again, we really appreciate your participation in these polls. It helps to get to know you a little bit better. All right. Looks like we almost have all of our responses in. So without any further delay, I am going to bring Paris on to join me. Good morning.
Hello.
Kick things off. Thank you.
All right. Well, thank you, Sydney, for that introduction. Once again, my name is Paris. I'm a PhD candidate wrapping up my work at the University of Guelph in Canada. And I wanted to start by thanking the American Physiological Society, the Society for Women's Health Research, and Insight Scientific for organizing this fantastic women's health webinar series, and for Aurora Scientific for inviting me to speak here today. It's a privilege to participate in this important platform that highlights research in women's health. And today I'll be discussing the effects of ovarian failure on muscle form and function. And I say ovarian failure because that's our model of menopause, which is not just a pause in menstruation, but rather it's a natural and significant transition in a woman's life. And it occurs when the ovaries cease to produce mature eggs for ovulation, leading to the end of menstrual cycles. And this is usually diagnosed after going 12 consecutive months without a menstrual period. The main hormonal shift involves a decrease in estrogens and progesterone, and these changes are the primary drivers behind most of the symptoms associated with this transition. Here we have levels of estrogen throughout a female lifespan in purple, and we can see that by age 40, there's relatively normal levels of estrogen. And what's not shown on this graph is another hormone called follicle stimulating hormone, and we have normal levels of FSH around this age as well. And I'll come back to the role of FSH in the ovary. And then if we look at age 50, there is a significant reduction in levels of estrogen. And again, what's not shown is that there's an increase in FSH levels. So what's happening in the ovary here is, well, FSH is produced in the brain in the anterior pituitary gland, and it travels from the brain to the ovary, the female gonads, where it binds to its receptor on these primary and primordial follicles. We actually have, we're born with millions of these, and there's a finite amount of these. But by the time we're in, we hit puberty, we have a few 100,000 left. So FSH arrives in the ovary and it binds to its receptors on these primary and primordial follicles, which causes the maturation of these follicles, eventually to the production of a mature egg that is released for ovulation. And in the process of this maturation, the secondary and tertiary follicle stages have what are called granulosa cells, and these are our estrogen-producing cells. So we can say that FSH's role in the ovary is follicle maturation and indirect estrogen production. Another type of cell we have here are theca cells or theca casts, which are our androgen or testosterone-producing cells. Now, what happens post-menopause is that we've depleted or exhausted the primary, like I mentioned, we have a finite amount of these. So we've depleted our primary and primordial follicles. So there's no receptor for FSH to bind to. So it arrives in the ovary, but it cannot cause the maturation of these follicles, and thus we lose the capability to produce estrogen in the ovaries. However, we do retain our theca cells, so our androgen production is not halted post-menopause. And here we have a hormonal profile of what our hormones would look like across a lifespan. In green, we have FSH, and in purple, we have estrogen. And relatively, estrogen is fairly consistent and high for most of our reproductive years, and FSH is low until it starts to fluctuate a little here. And then around the age of 50, when menopause sets in, estrogen arrives at an all-time low and FSH at an all-time high. And that's because there's a negative feedback loop between these two hormones, because the brain will try to produce FSH, send it to the ovary to produce more estrogen, and it will constantly receive feedback that that command was not carried out. And then the ovary eventually becomes desensitized to this FSH. So no matter how much FSH is reduced and the levels keep rising, the ovary is not capable of producing any more estrogen. Now, this transition period is quite interesting. We can see that it can take up to 10 years for females to become menopausal. And these fluctuations in the hormone come with an abundance of symptoms. And this is a phase we call perimenopause, which is essentially the runway to menopause. And like I said, it often begins several years before menopause, can take up to 10 years in some women, and is marked by less predictable menstrual cycles and initial symptoms. And why we're interested in studying the perimenopause phase? Well, throughout history, women have fairly been underrepresented in research, but at least the past few decades have tried to make up for that a little by focusing more on the reproductive years. And less so than the reproductive years, but still a growing body of literature is becoming more and more available now on the menopause phase of a woman's life. So definitely a huge step in the right direction. However, perimenopause, this poorly defined period of time spanning from the first incidence of menstrual irregularity to 1 year after the last menstruation, describes this transitional phase between pre and post menopause. And we don't know much about this transition phase. And yet we know from results from some big studies like the Women's Health Initiative, one of the largest female studies in the United States to look at menopause to date, that it's important to understand what's happening in this perimenopause phase. Here is what menopause can look like in women. There's definitely a large variety of different symptoms that women unfortunately experience with this transition. And recognizing these symptoms early can help in effectively managing them. And my interest, of course, here lies in muscle function and how menopause or ovarian failure affects muscle function. I think muscle function is particularly important. Here I have a graph that has age on the x-axis and muscle mass and strength on the y-axis. And this is pooled across both sexes, males and females. And we see that there's a loss of muscle mass and strength with age. And this can make it harder to carry out daily activities, leading to a lower quality of life. It can also increase the risk of falls and fractures, which can also lead to a loss of independence. Now, here they have it labeled as disability. I'm going to call it a functional threshold. There is a functional threshold needed to perform activities of daily living. And when you drop below this functional threshold, you're at a risk for not being able to perform activities of daily living. And if you're maintaining a healthier lifestyle with exercise, then this might be the drop in muscle mass and strength. So just to review, this is pooled between for both males and females. But if we wanted to separate them and look at strength, and specifically strength of the knee extensors, as Samson et al. Have measured here, we can see that around the 5th decade of life, women have a faster drop in strength compared to men, which happens to coincide with the age at the onset of menopause, which I find really, really interesting. And we know from other literature in humans that it's not just a loss in strength, but it's also a decline in muscle mass, as well as metabolic changes, which often lead to increased body fat, which ultimately will affect our muscle function. And all of this happens because of a lack of estrogens in the body, which play critical roles in maintaining muscle mass and function, and they regulate a lot of different metabolisms like repair and regeneration. So just to go over a few common methods of how we would study menopause in humans and how it affects muscle health or muscle function is we would have to run what we would call clinical studies or longitudinal studies. And we'd have to control a lot of factors like age, time since menopause, which is proven since the early 2000s to be very key, exercise history, medical history, or family history, and other symptoms experienced. It's pretty hard to control for all of these factors, and it limits our recruitment pool. And if we were interested in assessing functional changes in muscle strength due to menopause, we would likely use whole body dynamometry. And if we wanted to determine changes in muscle activation and fatigue rates in postmenopausal women, we'd likely pair this whole body dynamometry with electromyography analysis or EMG analysis. If we cared about more cellular or molecular changes in muscle tissues, or if we wanted to identify biochemical changes linked to muscle degradation, we'd likely use a biopsy technique where we take a little bit of muscle from the human and analyze that, or we could run biochemical assays. And lastly, if we wanted to look at muscle atrophy, for example, or fatty infiltration, we'd use imaging techniques like ultrasound and MRIs. Now, while human studies are fantastic, and they've been fantastic at telling us what the consequences are, they're not always the best for telling us the why. We often need more mechanistic approaches or methods to try to answer some of the whys. And this is where animal models typically serve as a great tool to study menopause. They give us a lot of flexibility, and we have more control over various factors that we don't normally have control over when we're looking at human studies. So I'm just putting up this human menopause profile, hormonal profile, once again, just as a reference. And I'm going to start talking about the first animal model, which is a rodent reproductive senescent model. And rodent, which is a natural aging model. So we wouldn't induce menopause in this model. We would allow rodents, female mice or rats, to age, and they go through their version of menopause, which is called estropause. And particularly in aging female mice, estrogen levels can even rise due to continued sporadic ovarian activity and a lack of a definitive shutoff. And unlike in human menopause, where estrogen levels remain low after ovarian failure, rodent estrogen can fluctuate and even spike, actually, likely due to an accumulation of anovulatory cycles or cycles without ovulation. Which would still involve follicular development and estrogen release. So this model is used normally for aging, but it's not fantastic for drawing similarities to human hormonal profiles. The next model is a rodent ovariectomy, or OVX model, that I want to discuss, which involves the surgical removals of both of the ovaries. So here in the senescent model, the ovaries of the rodent is intact, but in the OVX model, we do a surgical removal of both ovaries, which causes an instantaneous loss of circulating estrogen levels, which contrasts with the gradual hormonal depletion that occurs during natural menopause. So we see here we're not able to capture the perimenopause phase that much with the rodent ovariectomy model. So this model implements an abrupt and complete estrogen loss, providing sort of a worst case scenario for studying the effects of estrogen deficiency on tissues such as muscle. And it is useful, however, for examining the immediate consequences of estrogen withdrawal, such as muscle atrophy or increased fat deposition in the muscle. And before I move on to the next model, I just want to show that studies using the OVX model in mice have demonstrated that estrogen deficiency leads to significant reduction in skeletal muscle mass and function. And these results that I'm showing here from Moran et al. Are from the Dawn Lowe group, who publishes a lot in the field, and I actually base a lot of my research on hers. So in this study in the left, they investigate a slow type muscle, which is the soleus, and a fast type muscle, which is an EDL. And I'll come back to what the slow type and fast type are, but they include different fiber twitch properties in each of the muscle. And soleus is known more for its slow twitch properties, and EDL for more of its fast twitch properties. And when they did this study, they found they did it first in 2006, and they showed that there's a reduction in force. So there's force here on the y-axis. And in a follow-up study, they reintroduced estrogen, and those are the hashed bars here, and saw that force had recovered. So the force loss that we see here in the white bars in the OVX was recovered when estrogen was reintroduced. So we know that estrogen plays this important role in the regulation of force output in muscle. Now, this reduction in strength is generally attributed to a decrement in the fraction of strong binding myosin during contraction. And conversely, treatment with estrogens increases strong binding myosin and ultimately strength. So I'm just going to touch on what these two measures are briefly before we move on. The strong binding myosin is in the EDL that they have here is this is studied at the single fiber level, and active stiffness here that they're reporting is at the whole muscle level. And generally, the fraction of myosin in the strong binding structural state during contraction directly reflects force generating capacity, and active stiffness of intact muscle indirectly reflects myosin strongly bound during contraction. And both of these measures are affected by estrogen status. Which brings me to the last rodent model that I want to talk about today, which is called the rodent AOF model, or accelerated ovarian failure model. And for the rest of this talk, I'm actually just going to refer to this as the VCD model, because we use a chemical called VCD, which stands for 4-vinyl cyclohexene diapoxide. VCD is fine. And it's used in rodents to simulate gradual ovarian failure, which closely mimics hormonal decline seen in human menopause. And it's important for me to highlight that menopause doesn't start with a light switch like we've reviewed. It's a process that occurs over time. And this model allows us to study that process, and it allows us to assess time course changes. And this is something that the other models don't exactly allow for. And more importantly, as opposed to the rodent ovaryectomy model, where the ovaries are surgically removed, here the ovaries remain intact. And how VCD works in the ovary is when we treat the mice or rats with VCD, this chemical that selectively destroys those primary and primordial follicles that we have in the ovary. And over time, this leads to an accelerated gradual depletion of these follicular reserve that we're, the finite number that we're born with, which results in a diminished estrogen production and eventual ovarian failure, resembling this perimenopausal transition that we see in women. So the process of atresia, which is the gradual depletion of ovarian follicular reserve, which happens in human menopause as well, is accelerated in this model. And that's what's interesting, is that it makes it just like human menopause, these theca casts or theca cells that are still working in the VCD model. So we don't lose our androgen producing cells, not in human menopause and not by using this VCD model in mice. And I just want to add that this is probably as close as we've gotten to studying menopause in animal models, which brings me to why I decided to use this model for my thesis dissertation. So I wanted to use this model to look at how the gradual transition to menopause impacts time course changes of single muscle fiber contractility, and I'll get to that in a moment. But because this model provides a progressive reduction in estrogen that closely mimics natural menopause, it offers insights into how prolonged hormonal decline affects different muscle fiber types. So this is the whole muscle level. We have whole muscles that are attached to bone via tendons. And if we zoom into this muscle, we see fascicles that grouped together make up a whole muscle. And if we zoom in more, we see single muscle fibers that group together make up our fascicle. And if we zoom in one more time, we see sarcomeres, which are the most basic contractile units of our muscles. This is where all of the force production in the muscle occur. And we know in menopause that women become weaker at the whole muscle level. And this involves voluntary contraction of muscle. And there are a lot of factors that contribute to force production at the whole muscle level, like the ability to activate the muscle, the length of the muscle, the in-series compliance of the muscle, and a lot of factors that we can't always control for. What I'm doing in my research is that I'm studying this at the single muscle fiber level. And at this level, I can control all the chemical solutions, ions involved in muscle contraction. So I could create the perfect environment for the muscle to contract. And I do expect that with menopause, there will be impairments in muscle contractility at the single fiber level. And these are really unique properties that we can study at this level, at the single fiber level. Taking out neural control and impairments in nerve production, we're studying these actin and myosin filaments, which are the most intrinsic contractile properties of muscle. And in our whole muscles, they're made up of different muscle fiber types. You've likely heard of slow twitch fibers and fast twitch fibers. And the slow twitch fibers are more oxidative in metabolism, and they're better used for more endurance type activities because they have more access to blood and capillaries. And so when we look at them, they actually appear more red in color compared to our type 2s, which are more glycolytic in metabolism, and they're better used for explosive or strength activities. And if we wanted to compare what a fast type fiber looks like, or muscle looks like compared to a slow type, we would probably think it's more palish or pinkish in color. And so for my study, because we have this model now that allows for time course assessments, I wanted to look at how different stages of menopause are affected using the VCD model. So I've chosen 4 time points that I've tested my single fibers at mechanically, and those are the perimenopause stage, onset of menopause. So this would be considered day 0 of an OVX model if we were to do an ovarectomy, 2 weeks post-menopause and 8 weeks post-menopause. And I chose these 2 time points to try to reflect, tease out some differences that we might see at early stage and late stage menopause. So what we do is we bring these mice in, we acclimate them, and we inject them with VCD over 15 days. Now you can control the dosage of VCD that you want to do, and the dosage you give will impact the duration of the perimenopause phase. But we chose 15 days, and we ensured that they're in this diestrous phase, which confirms that they're in menopause. And we do this by using a vaginal cytology. And then for our time course assessments, we sacrifice some of these mice at perimenopause time points, some at the onset of menopause, some early and some late menopause. And then we harvested their soleus and EDL muscle. Soleus would be a representative muscle of our slow twitch and slow fibers, and EDL would be a representative of our explosive or fast type fibers. And then we chemically permeabilized these muscles, and I can talk about what that means later. I'm just going to jump over it for now. And we dissect single muscle fibers from these prepared bundles, and we perform mechanical testing on it, which is a really cool process. Here is our setup here, and I wanted to show you a close-up of what this looks like. So here we have what we call the Aurora single fiber system. And this is a specialized system designed for conducting detailed mechanical tests on isolated single muscle fibers. And the system is particularly valuable for studying muscle contraction dynamics, like cross-bridge cycling or calcium sensitivity, because one of its key features is its ability to switch between baths containing different reagents or ionic conditions, which is important because muscle contraction is dependent on calcium and ATP, which is the energy source for muscle contraction. So each one of these baths could hypothetically have a different solution. So I would normally put similar levels of ATP in these different 6 baths, but I would change the concentration of calcium to try to influence the strength of muscle contraction. So if a solution had a higher concentration of calcium, I would expect more force output from it. So higher calcium, more force. And I'd like to bring your attention to these pins here that I hope are clear enough for you to see. So this is a really tiny pin that measures the length of our single fiber, and this is attached to our motor arm here. And then this other pin is attached to our force transducer. It's seen over here. And this force transducer measures the level of force in our single fibers. And a more zoomed in version of this is here. Our single muscle fibers are tied between our length controller pin and force transducer pin. And just to appreciate how small a single fiber is, the thickness of a single fiber is basically as thick as a single strand of hair. So, learning this technique is quite challenging at first, but you can get some really cool data out of this. So we tie the single fiber to these pins via these sutures that we make in-house. And then, like I was mentioning, the Aurora system is equipped with this multi-bath setup that allows the muscle fiber to be exposed to different solutions. And by seamlessly transferring the fiber between baths, we can test calcium sensitivity, force generation, and this feature is especially useful for our permeabilized fiber studies where the intracellular conditions are completely dependent on the experimental setup and the solution. So the fiber contracts as well as we lay out the solution for it too. And I want to walk us through what one testing raw data trace would look like. Here at the bottom, we have length. So this is the length of the fiber, and this is an isometric contraction. So the fiber is not shortening or lengthening. The fiber is kept constant, aside from this super tiny length change here, which I'll get to in a moment. But the length is kept constant and isometric throughout the whole testing protocol. And up here, we have the force output. And at the start here, this is where our fiber is in that biggest bath here, where we usually put a relaxing solution here. So there's no calcium in this solution, and there's generally zero force that should be produced at this stage. And then this squiggly line represents the time where the fiber is moved out of the bath, moved and put into the next bath. And generally that's an activating solution with high levels of calcium. In this case, this is maximal calcium available. So the fiber starts to produce force, and this is what we call activation rate. And then a steady state is reached here, and we call it steady state because there's no longer climb in force, but rather the force plateaus. And here we can take our measurement. When I'm talking about absolute force measures in the next slide, this is what I'm referring to. I'm taking the force that is produced at this stage of my contraction, and then I could do a stiffness measurement. I'll come back to it when I'm discussing the result. We can do a slack re-slack method, which gives us an idea of rate of force redevelopment. And then finally, the fiber, the squiggly line again, is when we take it out of that activating solution and move it back and put it into the relaxing solution. So we can do a lot. We can get a lot of measures out with just one activation, which is kind of the beauty of all of this. So here we have some results, and all of this is result of my soleus or my slow type muscle. And I'll get to the EDL in a little bit, but just to bring your attention to the left panel here, this is our absolute force measure on the left y-axis. And we have menopause stage, those 4 different time points that I talked about: perimenopause, onset of menopause, early phase or 2 weeks post-menopause, and 8 weeks post-menopause, or our late stage menopause. And all of our red bars across all of these are our control animals that are age matched to our mice who are induced with BCD or are now undergoing ovarian failure. So absolute force is our measure of strength. And what we saw was that at the onset of menopause, the VCD fibers were stronger, and 2 weeks later, they were weaker. And 8 weeks later, they were stronger again. And anytime you want to talk about strength or force in a fiber, we have to consider fiber size, because what if the changes in fibers are because the fiber is bigger or smaller? You know, we can think of a bigger fiber being able to produce more force. And so that's what this measure here in the middle is. CSA is our cross-sectional area measure, and it gives us an idea of the size of the fiber. And so what we saw was that, at the onset of menopause, there is an increase in fiber size. So we can attribute that this increase in force that's observed here is due to a bigger fiber size. Our next, and we still don't see any differences for fiber size for our post-menopause time points. So then we're looking at our specific force, which is our strength measure divided by our cross-sectional area. And this way, it gives us an idea of muscle quality. So it takes into account fiber size and thus protein content in the muscle. So we see, using our specific force, with our specific force measure, that there's a decrease in specific force right at 2 weeks after menopause, and an increase 8 weeks after menopause. And our specific force can be closely tied to our instantaneous stiffness method. I mentioned I'd come back to it. This is a measure that reflects the proportion of attached cross bridges between those actin and myosins at any given moment. So this is instantaneous. And in single muscle fibers, it provides this insight into how many cross bridges are attached and contributing to force production. And here's how it's typically measured. We induce a small rapid, or we apply small rapid changes, and they're so small that they don't even show up on our length change here. But we can see that this is our stiffness measure, and these length changes are so small, on the order of nanometers, and they happen so quickly, milliseconds or less, that they do not allow time for new cross bridges to form or break. So the fiber's response to these changes directly reflects the stiffness of the existing cross bridges. And so the force response to these quick stretches or releases is measured. So when we look back here at our instantaneous stiffness, we see that at our 2 weeks post-menopause stage, we see a decrease in our stiffness, which means there's less bound cross bridges, and an increase in stiffness in late menopause, which we would think means more bound cross bridges. And if we compare these to our specific force data, then we can tie it back and say that, well, the decrease in specific force that we see at 2 weeks post-menopause can be attributed to less number of bound cross bridges, and an increase in force can be attributed to more number of bound cross bridges. And the next measure, and the last one before I move on to our EDL muscle results, is our KTR measure, which provides valuable information on the kinetic properties of muscle fibers, particularly how quickly they can generate force after disruptions. And it helps us understand differences in muscle performance under various physiological conditions. And how we measure this for KTR is the fiber is held at steady state, and that's here. It's in steady state of contraction, and then it is rapidly shortened or slacked by roughly about 15% of its original fiber length. And this shortening releases tension where the force drops to zero. Remember, I said this was our baseline where it's still in a relaxing solution. So the force here drops to zero, effectively disengaging the cross bridges between actin and myosin filaments. And after the fiber is shortened and the tension is released, the fiber is quickly returned to its original length or reslacked. And this allows the cross bridges to reattach and redevelop force. So the force generated during this reslack phase is recorded by our force transducer, that pin, and the rate at which force is redeveloped represents the rate of cross-bridge reattachment. And this type of force regeneration is different from our initial activation, because pre-activation here, like this force rise, is due to calcium being introduced in the cell, whereas here, pre-force rise, calcium is already maximal. So it's really just a reslack that is causing the force redevelopment. And this, so I was just mentioning that these two are completely different measures. And I said all of that only to let you know that we actually didn't see any differences in our KTR measures between any of the groups across any of our time point, other than our 2 weeks post-menopause group just had slower force regeneration compared to the other time points. But there was no differences between the groups. But I wanted to highlight that if any researchers are interested in looking at cross-bridge dynamics in cycling, that this is a really cool measure that they can use. And so here we're at our EDL muscle, and I've tried to use the same layout just to follow the same idea. So absolute force, cross-sectional area, and specific force. And a very interesting finding was that we actually didn't see any differences between our menopause or VCD groups compared to our control groups in the EDL muscle across any of our measures, which was different than what the OVX results in the literature indicate. And these might be for a variety of different reasons. You know, there's some research out there that says that slow type fibers have more higher density of estrogen receptors, which is how estrogen is able to interact with the muscle. And so they would experience the loss of estrogen deficiency faster, or they would be more impacted by that estrogen deficiency compared to EDL muscles, especially in this gradual ovarian failure model. So to quickly summarize, we saw no change in most of our isometric measures. So everything I've presented up until now, these were our isometric measures. However, when we look at dynamic properties of muscle, the story is different. This study was conducted and led by my colleague, Emma Hubbard, who's published this paper now in AJP Cell Physiology. And I've actually linked this in the handouts. And she only looked at one time point, which was a 2 weeks post-menopause, and we see differences in our power output measure. So our force measure, or the previous strength measure, is a static measure of force production. So the muscle is not moving or shortening during the contraction, whereas power has both a strength component and a velocity component, or a speed component, and is a measure that captures muscle function during movement. So both strength and speed can be affected differently across menopause transition. And in this dynamic measure, muscle fibers undergo shortening contractions under load, allowing us to assess how force and velocity combine to produce power. Power is force times velocity during active movement. And we saw in our results that there's a reduction in power in both the EDL and soleus muscles. So she actually also pooled, the gray bars here show the EDL muscle and the red are the soleus. So she pooled her muscles together for her analysis. And then we thought, well, is there any way that we can mitigate this power loss? And we thought maybe exercise would help. And we're still trying to investigate the effects of different types of training on attenuating impairments in muscle contractility. But one of the ways we did this in our current study, or in the study that's published, was by running mice uphill. And we did see that, yes, when we trained these mice, that power loss was attenuated. And if you're interested in seeing what that looks like, I have a little video here where we used a high intensity interval, or HIIT protocol, which involved short bursts of intense exercise followed by recovery periods. And this HIIT protocol was selected for its potential to counteract the negative effects of estrogen decline on muscle function. And what we did was we set the treadmill to a 25%, 25-degree incline, and then this training was repeated 3 times a week, Mondays, Wednesdays, and Fridays. And the mice ran at a base speed, and then they sprinted for 3 sets, and then they finished off with a dash. And the exercise was progressed bi-weekly through increasing intensity levels. So through the 8 weeks that they trained, they first started with a base speed of 5 meters per minute, and their dash was 10 meters per minute. And then by the last couple of weeks of their training, they actually had a base of 11 meters per minute, which is faster than even their dash when they first started. And they finished off with 21 meters per minute. So this was progressive training over 8 weeks. And just a quick recap before I move on to some other cool studies. We didn't see any meaningful differences in force isometrically between the two muscles. But when we looked at our dynamic measure, our power with the velocity component, we're starting to see big impairments in contractility. And, power is important because it's a dynamic measure, which is crucial for understanding real world muscle performance. Here's another view of what my specific force in the soleus looks like. On the left, this is our soleus muscle with all of its fiber types pooled together. And then here, I've separated them by fiber type as well. And this is work that is currently under review. But I believe I've sent the preprint version of this, and it's linked in the handout section if you're interested in looking at all the measures we had. But we fiber typed these fibers, and what was interesting, if I want to bring your attention to these dotted, the VCD groups, and we can see that there's more blue dots here in the VCD groups compared to the red dots. So is there a shift to more type 2 fiber types in the VCD model? It made me think, what am I missing at the single fiber level? What else could be going on in our muscles that with the VCD model that I'm not able to see with a single fiber model? And so what my next study, and this is something that's currently I'm working on, and so I unfortunately don't have data to show you, but I'm interested in looking at how different fiber types, slow twitch and fast twitch, change and are affected morphologically. So I'm going to look at these muscles. If this is what a control muscle looks like, is this what a VCD muscle looks like? Is there more fat? The yellow dots represent fat droplets, and the white lines here are increase in fibrosis. So is there more fat and fibrotic tissue? Is there more collagen content in our VCD model, VCD muscle? We don't know. There's a lot of studies that have looked at gene expression and upregulations and downregulations of fat oxidation genes, but we don't actually know what these muscles look like histologically, and that's what my next study looks at. So we repeated our VCD model where we injected our mice with VCD, and it takes a while for this model to develop based on the dosage that we give these mice. It takes almost 4 months for them to be menopausal. And we actually added in an estrogen group here as well to see, well, if our VCD group happens to have more fat and more fibrosis, then can estrogen reverse this to change what the muscle looks like back to its control state. And we confirm menopause just like we did with the first studies with vaginal cytology confirming diestrus. And this is when we would start them on estrogen, and we put estrogen pellets. We implant estrogen pellets in these mice. We allow 8 weeks to pass, so we were only looking at one time point for this study, and we harvest the soleus and EDL. We try to maintain all the same muscles used so that we can translate between our single fiber data as well as our histological data, and then also put that in the context of the literature, because these are the most commonly used muscles in animal models. And then we thought, well, okay, that's wonderful, but what if we have a control group and a VCD group, and we have some of these mice that are sedentary, some of these mice are put on estrogen, and some are put on uphill training on an exercise protocol to try to get at non-estrogenic pathways and whether we're able to reverse the state of the muscle back to its control state by using exercise. And then we allow the 2 months to pass, and at the 8-week mark, we decided we're going to put them on a high-fat diet, and this was to induce a secondary metabolic insult. The VCD is a primary metabolic insult. It goes in the muscle and supposedly messes up the muscle, and the high-fat diet here is supposed to exacerbate that effect. And this way we can get at some really cool mechanisms, like is our training and estrogen protocol, are they enough of a stimulus to reverse not only a VCD a group, but our high-fat diet VCD groups back to its control state. And then we allow 1 month to pass, so they're on this high-fat diet for about 4 weeks or a month. And then at the 12-week mark, we harvest the muscles and we look at them histologically. And here's kind of what we think that might look like. We have our control muscle. You know, if they're undergoing VCD treatment and they're deficient in estrogen, is this what it would look like? What if we pair VCD with a high-fat diet? Will they have more fat and more fibrosis? If they're just put on a high-fat diet, how does that differ from these other groups? And what if they're on VCD and a high-fat diet and we exercise them? How possible is it to get the state of these muscles back to the control? And likewise with our estrogen reintroduction. And so as a quick take home, we used the VCD model and we induced gradual ovarian decline or estrogen decline, which mimics natural menopause. And this allowed us to observe subtle but progressive impacts on muscle health that differ from the abrupt loss seen in OVX models. But really, the VCD model has provided a unique lens for studying menopause, enabling us to capture realistic hormonal transitions and better understand their implications for muscle performance in animal models. And also our isometric measures, well, they revealed distinct responses between muscles. While the soleus had showed differences between groups at various time points, the EDL muscle, however, did not exhibit similar sensitivity to estrogen loss. And then for our dynamic power measurements, they uncovered broader impacts with both the soleus and EDL muscles exhibiting power loss in the VCD group, which emphasizes the significant effect of estrogen deficiency on dynamic function. And notably, this power loss was mitigated through HIIT protocol, which demonstrates the potential for targeted interventions to preserve muscle functionality. And just to hit it home one last time is that dynamic assessments, like our power assessment, offer closer approximation of real-life functional activities. And while our isometric measures provide valuable insights on how maximal force is produced, understanding how these muscles perform in dynamic scenarios is essential for a more comprehensive view of daily movement capabilities. And my upcoming work on this morphological changes, stay tuned. I've also linked our lab website in the handout section, I believe, so that you can keep up with the latest work that come out of our lab. But my upcoming work will pair well with my single fiber findings in trying to get at whether this fat infiltration or supposed fat infiltration or fibrosis contributes to the observed changes and whether and we'll clarify how estrogen deficiency affects muscle structure and function at our single fiber level. Which brings me to my last point, which is yes, like this study was conducted at the single fiber level, which is great for isolating specific effects. But to understand completely, it requires examining how these fibers interact with each other and integrate with the whole within the whole muscles and the whole body. And future research should try to address these single fiber findings, translate and how they translate to muscle coordinated muscle action. And here I have a photo of a human single fiber, but I am testing this at the mouse level. So mouse single muscle fiber, which is a whole species removed. So translatability of all of these is important to consider when we read these papers and findings. Which brings me to my last slide. And I just want to take a moment to thank my lab members, my advisor, Dr. Jeffrey Power, who didn't say no to any of my projects, especially when I wanted to switch from mechanical testing to histology to try to answer some questions. And to my mentor and collaborator, Dr. Glenn Pyle, who is the person who taught me how to use the VCD model. He does work on cardiovascular disease and how cardiovascular health in women is impacted post-menopause, or how women are impacted post-menopause in their cardiovascular health. Of course, I'd like to thank the Aurora scientific team. Like I mentioned, this is all done at the single fiber level. There's a lot of things that could go wrong, but they are fantastic at always being available to help either virtually or coming in person to troubleshoot. And of course, my funding agency for funding my research. I've left my contact information here if you guys have any direct questions you want to ask me. And then I'd like to thank everyone who attended my talk today. I hope you enjoyed this presentation as much as I enjoyed putting it together for you. And with that, I'd like to take the remainder of the time to answer any questions that you guys might have.
Thank you so much, Paris. What an amazing talk. That was so interesting and insightful. So I'm going to give you a second, actually, before we start our Q&A to catch your breath and sip some water after that great talk that you gave. And just want to remind the audience to use the Q&A tab. We already have so many questions coming in. Please send in all of your questions. We're going to get to as many as we can live today. But if we do run out of time without answering your question, all of them will be sent over to Paris, and she will provide some written responses, and you should have answers to those within the next couple of weeks. So keep an eye out for that. Now, just before our Q&A as well, I have a couple more polls for everybody. So our next poll for today's session is, which of these techniques do you currently employ in your lab? So this is another select all that applies. And some options include histology, omic analysis, treadmill or wheel running, grip strength, wire hang, muscle function, or gait analysis. So go ahead and select as many of those that apply to your own research, and let us know what techniques you use for your own research. While you are answering that as well, I do want to just quickly leave a pop-up link for you to complete our post-event survey. If you have to leave early before the survey gets launched at the end of the webinar, you can click this link and just let us know your feedback on today's talk. Let us know, what webinar topics you might want to see from us in the future. And you can also submit those last questions in there if you do have to leave early. So yeah, we appreciate your input on your experience. Okay. And for our very last poll, what type or types of muscle preparations do you currently utilize in the lab? Another select all that applies. So our options here are permealized fibers, intact fibers, engineered constructs, whole muscle in vitro, in situ, or in vivo, or none of the above. And again, go ahead and select as many that applies to you there. Okay. Want to make sure we have enough time to get to some questions, and we may end up running a couple minutes over to make sure that everyone's questions, or as many questions as we can, get answered. So I'm going to bring Paris back on. Hi. All right, let's start our exciting Q&A session. I am really excited for this.
Cool.
Let's see. Okay, first question. Thank you for such a great talk. To what extent does the VCD drug model mimic menopause? For example, at what age are these animals treated relative to the age of menopause?
Okay, well, thank you for that question. That's a very important question. I wish I had a little more time to expand on this during the presentation, but I'm more than happy to here. One of the great benefits of using animal models is controlling many factors, like I said. And one of the factors that contributes to menopause is age. And so what we did for this model is we actually removed the aging component. So we induced a menopause. So we injected mice with VCD at a younger age. So these mice are sexually mature young mice. They're not at all near menopause or estropause for these mice. And we did these so that we can focus on what are the actual changes that occur in the contractility of the muscle without the aging factor. So these mice were, let me see, probably a few months old. They were around 2 to 3 months old upon arrival. And when we started their VCD treatment, they were just under 4 months old. And then by the time they were fully menopausal, they're around 8 months old, which translates to about like 20 years old in human years. And I think, was that the question? Did I answer?
I think so.
Okay,
And we can always go back, like I said,
cool.
when I
Yeah,
send you the questions, no worries.
sounds good.
Um, do you notice any difference in lean mass and muscle weight after menopause?
With the model I've used in this, and I've done this 4 times now, um, uh, and the last time was the only time that I've measured muscle weight and mass and length of the muscles, and I haven't analyzed the data yet. But from preliminary results, I don't think there's any difference in lean mass or weight, and this might be because of that whole aging thing. These are fairly young mice, and if I had to speculate and there were any changes in mass, it would actually be increasing because they're in that still maturation age, like they're growing, and that's why their muscles would weigh differently. But I'm more than happy to take a closer look and send you a direct response.
Wonderful. Okay, great. Um, our next question starts with fantastic presentation, and I have to agree. Do you know if the levels of myosin, actin, and estrogen receptors, alpha, beta, or GPR30, change in soleus and EDL muscles after VCD treatment?
Unfortunately, I don't know if that happens, and in fact, I don't think it's out there. I don't think it's been measured. The VCD model is fairly new. It's been used for Alzheimer's disease quite a bit. It's been used for cardiovascular health and disease, but first muscle contractility, very, very little. I want to say we're probably the first who are doing this in using this model in skeletal muscle. So I definitely don't know. I can take a look, but I doubt that this information is out there.
Yeah, potentially something to look into
Mhm.
next. What is your rationale for only treating the animals with VCD for 15 days instead of extending the treatment window to further accelerate ovarian failure?
Very, very cool question. So the amount of VCD that's given to these mice or injected will dictate how fast they undergo ovarian failure. And the 15 days of consecutive, so this is daily IP injections, uh, paired with the dosage that we gave them, models um, the ovarian failure and the perimenopause phase to match the human period of perimenopause phase the most closely. So yes, a lot of different labs who use this for different areas actually have played with the duration of injections to try to induce it faster or slower. So it all depends on your research question and what you're trying to answer. I was really trying to maintain as much translatability as possible to the human menopause. So I tried to follow the same dosage that would hypothetically reflect the same duration of ovarian failure or perimenopause. And that's why we did the 15 days with the dosage that we did.
Great response. That makes a lot of sense. Which strain of mice do you use for studies, and are there strain specific effects like genetics on the muscle responses?
We used CD1 female mice, and these are sexually mature mice that we order. And are there strain-specific effects? I don't think so. I don't think so. Other types of mice have been used by other labs. I don't think there's any strain-specific or genetic effects on muscle responses. We just use this because they're, at least this is a generic strain that's used for a lot of different types of models, and they don't develop tumors, or they don't develop any types of cancers or anything. They were less likely to. That's why we use the CD1 mice.
Makes sense. Thanks. Great talk. Do you know if physical exercise modulates estrogen levels perimenopause or postmenopause? Because estrogen can be produced by other tissue like adipose postmenopause.
Estrogen can actually be produced still locally in our muscles as well. Just like you mentioned, it is produced in other tissues like adipose. But because we maintain our androgen producing cells, this is so important. This is why I love this model. It's so similar to human menopause. We're able to convert androgens via this enzyme called aromatase that we have in our liver, we have in the brain, we have in adipose tissue and skeletal muscle. We can convert androgens to estrogen. So we can still locally produce estrogens, but it is at way, way lower levels than if we were premenopausal. And physical activity would modulate estrogen levels a little bit, because there's a cycle of the more you exercise, the most androgens you produce, and the more androgens we have in the muscle, the more can be converted to estrogens. And so there's a cycle that I would imagine would be, yeah.
Okay, great. We are at the top of the hour, but I hope, do you have a couple more minutes? Because we do have
Sure.
so many
Oh,
questions.
yeah,
I'd love
sure.
to keep going for maybe a couple more questions.
Sure.
Okay, great. The next one is asking, well, they're curious about any challenges you've faced using VCD. In your upcoming study, are you planning to treat them every Monday, Wednesday, Friday for 4 months?
So we didn't, good question. We didn't necessarily face any issues with our VCD implementation. The mice handle it fairly well. It's not a very stressful um, uh, thing for them. We do acclimate the mice too. So from their arrival until we start our injections, we handle them quite a bit so that they're used to being held, so that this doesn't produce a stress response at all. Can you repeat the second half of that question?
I think they were just asking specifics about your plan for your upcoming study.
So yeah, are we going to inject them Monday, Wednesday, Friday? Was that what they asked?
Yeah.
Okay. Well,
Know
no,
how many
VCD happens
detail you wanna
15
know.
days right at the start. We don't take any days break, no weekends off or anything. It's 15 consecutive days of IP injections, and then we just let the mice sit for our dosage. It's 4 months. They just sit for 4 months until they're menopausal, and then that's when our timeline starts, and that's when we start our thing. The Monday, Wednesday, Friday was for our exercise regimen. So we exercise them 3 times a week for a total of 8 weeks. And in my upcoming study, one is 8 weeks and the other is 12 weeks. And the estrogen pellets are inserted at the onset of menopause, so 4 months later, and it's just a surgical implant that's inserted. So they remain in the mice all the way from onset of menopause until the sacrifice date. And the estrogen pellet releases a daily dose of estrogen by itself. So it's just an implant that's left in the mice.
Excellent. Okay. Thank you for clarifying.
Mhm.
Would you expect to see a recovery of these contractile function impairments in your VCD model paired with hormone replacement therapy? Not sure if you can give any of your thoughts on that.
Yeah, but the thing is, I don't think we saw any force loss or any detriments. So originally, I was going to plan a training study as a follow-up to this to try to see if we can mitigate the force loss, but we didn't even see force loss. So we didn't do the training study to try to mitigate the force loss, which was really interesting finding. I suppose that the whole age thing will probably have a big impact. Age compounded with estrogen deficiency is probably what causes a lot of the heavy symptoms that we experience at menopause.
Okay, wonderful. I think we're going to go to one last question. We have still so many, which is amazing, but yeah, we just don't have enough time in the day. So for this last question, would you be able to speak as to how or why specific force and stiffness recovered at 8 weeks post-failure compared to the 2-week post-failure time point? Is the 8-week time point different than pre-levels?
So just briefly, but I would love to delve deeper in this in my written report. But what I didn't present here are my calcium sensitivity results. And what I saw in those calcium sensitivity tests was that at the 8-week time point, calcium sensitivity was improved. And so that would pair with the increase in force output that we see. So if you're more sensitive to calcium, that means you require less to produce more force. It means you're better able to form cross bridges. And so I would speculate that that's why. But like I said, I'd like to delve a little deeper in my report.
Yeah, absolutely. And as I mentioned, we're going to send all of your questions, audience questions, over to you, Paris. So, yeah, you can go as detailed or as brief as you'd like in the answers. And hopefully that helps everybody get the answer to their question, because it sounds like everybody was very curious and you did a wonderful job today. I want to say thank you so much for your time and expertise. And it was such a pleasure to have you with us. And I hope you had fun today.
I did. Thanks so much for that, Sydney. And thank you, everybody.
Yeah, you're very welcome. All right. And of course, thank you to our live audience. We know you have busy schedules, so we really appreciate you being here live, and I hope you enjoyed your experience today. Please join us for the next event in the series on Wednesday, November 13th at 11 a.m. Eastern Time. You will receive a reminder email with the link to log in soon. Make sure you block it off in your calendar so you don't miss it. Quick reminder, a recording of today's webinar will be available soon, so keep an eye out for an email that will give you access to that. Additionally, that survey will appear at the end of the webinar. We really do read all of your comments and appreciate your input on your experience today. And you can also let us know what webinar topics you'd like to see in the future. Finally, as I've mentioned several times, you can still submit your questions for our speaker and into the survey, and we will be passing them on to Paris. In closing, we hope you enjoyed this Inside Scientific webinar sponsored by Aurora Scientific. Another quick thank you to all of our partners for helping make this series possible, and I hope you all enjoy the rest of your day.